Thank you for your interest in Transposagen's gene editing technologies. This presentation will discuss footprint-free gene editing in cell lines for therapeutic and research and development purposes. The first component to the footprint-free gene editing system is the latest site-specific nucleases. These include TALENs and the recently engineered dimeric NextGEN CRISPR, shown here on the right. They each consist of a DNA binding domain fused to the FOC1 endonuclease, the enzymatic portion which is responsible for cutting DNA. Both nucleases shown here require the precise dimerization of the FOC1 domain to function. These site-specific nucleases have many advantages, which lead to a superior gene editing process. They are affordable, quickly assembled, have high efficiencies without toxicity, broad usage, and are incredibly specific to their intended gene target. If you combine these nucleases with Transposagen's piggyBac technology, the result is the most accurate gene editing system available today. Let's see how it works. We first start by designing NextGEN CRISPRs or TALENs close to your intended target gene. These will induce a double-strand break which will stimulate the homologous recombination pathway. By providing a donor vector, the cell will repair the double-strand break, incorporating the selection cassette and the desired change to your gene. The selection cassette consists of a positive and negative selection system, flanked by piggyBac ITR sequences. Using positive selection, cells with the incorporated sequence will be screened out. Only those with the cassette will persist. Following this process, excision-only piggyBac will recognize the ITR sequences and scarlessly remove the marker, leaving behind a correctly edited cell. A final round of negative selection will guarantee only cells without the cassette remain. So what can we use this for? Footprint-free gene editing allows for single base pair editing and is well suited for stem cell engineering. With this, disease-specific lines or isogenic controls can be quickly made. This type of work can lead to cellular therapies as well as treatments for a broad range of genetic disorders. There are some key advantages to the system. The process is highly efficient, allowing the change of both alleles in a single step. The high efficiency reduces costs and timelines. piggyBac itself is not mutagenic. It does not alter, insert, or delete genomic sequence. This is the only clean gene editing system with the ability for positive and negative selection. A very recent therapeutic application of our footprint-free gene editing system was published in Genome Research. Here is a gene correction of an inherited blood disorder, Beta-thalassemia, in patient-specific iPSCs. Researchers were able to use the system to quickly identify precisely edited clones containing the desired changes, saving both time and resources. This application can be applied to an unlimited number of other genetic disease models, including cystic fibrosis, Huntington's disease, muscular dystrophy, sickle cell anemia, and immune disorders. With our footprint-free gene editing system, Transposagen can provide the most technically advanced cell line engineering services. We have over 10 years of experience in project design, transfection, and cell line engineering. To date, we have worked with over 100 different cell lines. Our engineering services include every variety of gene edit available, ranging from a gene knockout to complex conditional systems. Our flexible, milestone-based services are budget-friendly and allow you to monitor progress closely. In summary, Transposagen can apply the latest technologies to deliver your cell lines with the most precise gene edits. Our technologies have already proven valuable to the research community, especially to those working with cell-based disease research models. Lastly, Transposagen has an abundance of cell line engineering experience and has flexible pricing options to meet your needs. We hope that you now have a better understanding of the gene editing possibilities with Transposagen. For more information on our cell line engineering services, please contact us by phone or email to get in touch with your local gene editing specialist.